Good evening to everyone and welcome to today's wonderful evening with Dr. Murali Bharadwaz. Every day we are meeting on the WhatsApp study group. Whenever I get time, away from the patients, away from the hospital, away from the clinic, I take every opportunity to spend with audio mantras. I post the questions along with the audio answers and try to engage you, stimulate you to prepare for the exam. And every day we will have at least 1 hour of live discussion together and solve the PYQs. So can the online students can punch the voice loud and clear for all of you, doctor? Let's make the great beginning. Can the online students can punch? So, a child with urine turning black. Yeah, good to see 7 online classmates. Yes. Please post in all your social media groups that the live class started along with the link so that everyone can be able to join. A child with a urine turning black on standing is diagnosed with alkaptonuria. So you should remember the urine turning black, and there is a deposition of the homogenesic acid in the cartilages, leading to the black coloration of the cartilaginous structures in case of the alkaptonuria. What are the most sensitive screening tests of the vitamin A deficiency? It is the retinol level less than 10 is considered to be the sensitive test. In thiamine deficiency, what do you see, doc? We see trans ketolase levels. So you should know, typically when the glucose undergoes a pentose phosphate pathway, it forms ribulose phosphate that become ribose 5-phosphate converting into glyceraldehyde 3-phosphate, where trans ketolase is very, very important. So this trans ketolase activity is an indicator of it has vitamin B1, thiamine as a cofactor. That's the reason trans ketolase is the one which is the enzyme assessed in to know the vitamin B1 deficiency is what you need to remember. So good to see 9 online classmates. Keep punching your comments, your thoughts, everything, doctor. That is how we need to enrich our preparation together. Right, doctor? Now, Keshan's cardiomyopathy is because of the deficiency of selenium. Zinc is very important. For a lot of things, zinc is a cofactor, especially enzymes like carbonic anhydrase, which will convert CO2 plus H2O, gives rise to H2CO3, which is broken down into H plus and HCO3 minus. For all these reactions, you require carbonic anhydrase, for which zinc is the cofactor. Whenever you heat, the protein loses its secondary, tertiary, quaternary structure, but not the primary. What is primary? It is all the arrangement of the amino acids is called as the primary structure. Zinc, copper, and iodine, they're all micronutrients, not the calcium. You need to know the list of the micronutrients. Urea cycle, where does it happen, doctor? It happens in the mitochondria and cytoplasm. Urea cycle involve both the some enzymes are there in mitochondria, some enzymes are there in cytoplasm. So this clearly says you should know everything, anything about the urea cycle, etc., etc. So, doctor, if you look at the urea cycle, right? So, carbon dioxide plus ammonia combine to form carbamyl phosphate. Carbamyl phosphate. For that, what do you require? Carbamyl phosphate synthase 1. Typically, ornithine transcarbamylase will integrate the carbamyl phosphate into ornithine to lead to formation of citrulline. Citrulline become arginosuccinate, arginosuccinate become arginine, arginine become ornithine. In the process, urea is released. So, the aspartate comes inside and combines with citrulline to form the arginosuccinate. So, arginosuccinate synthetase, arginosuccinase, arginase, they're all fundamentally located in the cytoplasm. Whereas the carbamyl phosphate synthase 1, ornithine transcarbamylase, they're all the mitochondrial enzymes is what you have to remember for the tomorrow's exam. Unconjugated bilirubin is a physiological uncoupler. So you should know electron transport chain. What are the various uncouplers? You have chemical uncouplers, physiological uncouplers. So 2,4-dinitrophenol, aspirin in high doses, it all acts as an uncoupler. What will happen if there is an uncoupling, doctor? You should know the phenomena. Whenever there is an uncoupling, then all the energy produced will not convert into ATP inside the mitochondria in the electron transport chain. All that gets converted into heat instead of converting into ATP whenever an uncoupler is there. So that is the reason whenever you take aspirin in very high doses, it being a chemical uncoupler, it leads to generation of a lot of heat, and that is the reason salicylate toxicity leads to hyperthermia. Is what you need to remember. Then there are some physiological uncouplers like thyroid hormones, long chain fatty acids, unconjugated bilirubin. Right? ये खाना खाने से पूरा गर्मी बन जाएगा बोल के, you heard it a lot, right? So what is the meaning of that? How will the temperature will rise if you eat certain foods? Because long chain fatty acids, unconjugated bilirubin, thyroid hormone. That's the reason in hyperthyroidism, because thyroid hormone is a physiological uncoupler of the oxidative phosphorylation. That's the reason it leads to excessive amount of the heat is what you need to remember. So these are all the PYQs, doctor. Every day we will do around 100 PYQs revision. Now, what is the principal building block in fatty acid synthesis? It is the acetyl-CoA. So you should know acetate and pyruvate, they lead to formation of acetyl-CoA, which in turn will go and form the fatty acids. For the acetate to become acetyl-CoA, you require acetyl-CoA synthetase. Acetyl-CoA synthetase. Whereas pyruvate to become acetyl-CoA, you require pyruvate dehydrogenase, for which vitamin B1, thiamine, is the coenzyme, is what you need to understand. Very nice to see 8 online classmates. Can you please punch the voice loud and clear for all of you, doctor? Lesch-Nyhan syndrome. What are the features of it? It leads to urolithiasis, gouty arthritis, because of the production of the urate. So you should know there is a problem in case of Lesch-Nyhan syndrome. Ribose 5-phosphate become 5-phosphoribosyl pyrophosphate plus glutamine. That become 5-phosphoribosyl 1-amine, ultimately leading to the formation of inosine monophosphate. Inosine monophosphate should become hypoxanthine. Hypoxanthine should become xanthin and uric acid. But this hypoxanthine also can lead to IMP formation, and inosine monophosphate in turn will form guanosine monophosphate and adenosine monophosphate. In this process, you require HGPRT, hypoxanthine guanine phosphoribosyl transferase. If this is deficient, hypoxanthine cannot become IMP. Hypoxanthine becomes xanthin and uric acid. That's the reason there is a hyperuricemia in case of hyperuricemia in case of Lesch-Nyhan syndrome, where the de novo pathway is affected because of the deficiency of hypoxanthine, guanine, phosphoribosyl transferase needed for xanthin. Hypoxanthine to become IMP, it cannot become IMP. Hypoxanthine become too much. Hypoxanthine becomes xanthin. Xanthin become uric acid. That leads to hyperuricemia is what you need to remember. So this is one of the age-old question that you need to remember, doctor. What is the initiator codon in prokaryotes? AUG. So you have start codon, stop codon. Kahan shuru karna, kahan rukna. The most common start codon is AUG. And the start codon in eukaryotes always codes for methionine, and the prokaryotes it produce modified methionine that is called formyl methionine. What will AUG will code for, doctor? In the case of prokaryotes, it codes for formyl methionine, whereas in case of the eukaryotes, it codes for methionine, is what you have to fundamentally understand. So what is the correct PCR sequencing? First, denature the DNA, anneal the primer, extension of the primer. That is a typical sequence which you need to remember. So this is how first you denature the DNA, anneal the primer, extend the primer. This is the typical PCR. Hot nuts disease can present with pellagra-like symptoms because of the niacin deficiency. So what you are seeing here, doctor, you are seeing all the skin manifestations of the niacin disease, niacin deficiency. Prostaglandins, from where are they derived? They are derived from the arachidonic acid. So the membrane phospholipids will give rise to, through cyclooxygenase pathway, thromboxane 2, prostacyclin, prostaglandins. So you should know, thromboxane 2 will cause the platelets to aggregate and form thrombus. Prostacyclin PGI2 acts on endothelium. Endothelium, it's a vasodilator. Whereas prostaglandin PGE2 is also smooth muscle relaxant. But PGF2 alpha is a smooth muscle contracted. And the lipoxygenase pathway will convert into the leukotrienes is what you have to remember. Mental retardation is a clinical feature of phenylketonuria. For the melanin synthesis, what do you need? Tyrosine. What are the 3 amino acids that are there in collagen, doctor? Proline, lysine, glycine, proline, lysine, glycine, proline, lysine, glycine. What is the specialty of proline? Proline is the amino acid which is called amino acid, because typically there is a ring structure, amino acid. So proline, lysine, glycine. Glycine is the smallest amino acid. Whenever any protein need to take a turn, then in all those turning points, you have the smallest amino acid, glycine, which will fit into it. So you should know, doctor, this very interesting point. What are the amino acids in collagen? 50% of amino acids are glycine, proline, hydroxyproline. So in order to make the proline become hydroxyproline, you require vitamin C, which causes hydroxylation. That is the reason in scurvy, because there is no vitamin C, there is no hydroxylation of proline, and because of that, the collagen becomes a weak collagen, is what you need to remember. Right, doc? Now, disulfide bonds. Can the online students can punch? Is the voice loud and clear for all of you, doctor? We have 1 hour May, 100 PYQs. Totally 22,000 PYQs. We need to do a revision in 220 hours. By that time, you will know what to read, what to focus, what to revise for the tomorrow's NEETPG INICET exam. So preparation for the NEETPG or FMGE is not something, oh my God, kind of a thing. You need to master, you need to conquer these 22,000 PYQs. That is all the beginning, doctor. Be very, very sure about it. So in cysteine, we have the disulfide bonds. So you have cysteine, cysteine. So SH are the disulfide bond in cysteine. Only SS without H is cysteine. They are the two amino acids which have sulfur disulfide bond. Menke is kinky hair disease due to deficiency of copper. G6PD, where do you find this? This is an enzyme on the membrane. So you should know, doctor, every enzyme is a marker of one of the organelle. Like plasma membrane, 5-nucleotidase, endoplasmic reticulum, alpha-glucosidase, cytosol, LDH, Golgi apparatus, galactosyl transferase, lysosomes, N-acetyl beta-glucosaminidase, mitochondria, malate dehydrogenase, inner mitochondrial membrane, succinate dehydrogenase and ADP synthase. Mitochondrial outer membrane is monoamine oxidase. Mitochondrial matrix marker is citrate synthase. Peroxisomes, it is catalase. Nucleus, it is lamin A and lamin C is what you need to remember. So mainly, doctor, you need to know typically what are the markers. Monoamino oxidase and voltage dependent anion channel, they are the markers of mitochondrial outer membrane. ATP synthase and cytochrome C oxidase, which is complex 4, is the marker of mitochondria on the inner membrane. And citrate synthase glutamate dehydrogenase is the marker of mitochondrial matrix. Hexokinase, pyruvate kinase, G6PD, they are all in cytosol. Endoplasmic reticulum mein, smooth mein, glucose 6-phosphatase, cytochrome P450 is present, is what you have to ultimately understand. So, doctor, G6PD deficiency, what is the type of inheritance? X-linked inheritance. What are chaperones? Chaperones provide a template how the other protein should fold. The protein folding is being controlled by chaperones. So whenever protein synthesis happen, it is all unfolded. Whereas whenever a chaperone comes in, it will help the folding of the proteins very fast, and they are removed for the usage. Melatonin, serotonin, niacin, they are all tryptophan derivatives. Amino acid tryptophan is the source of melatonin, serotonin, niacin. So tryptophan has a lot of functions. Tryptophan is a glucogenic amino acid. Tryptophan is a lipogenic amino acid. Tryptophan is 5-hydroxytryptamine leading to niacin formation. Then melatonin formation, indole acetic acid, proteins, they are all derived from tryptophan. Cytosine, adenine, guanine are there in DNA, but uracil is there in the RNA. Zinc is the one which is known to stabilize the secretion of the insulin. And where do you find cardiolipin? Cardiolipin is there on the inner mitochondrial membrane, cardiolipin location. A white odorless compound which lead to toxicity in a child, most likely it is iron, is what you have to remember. So doctor, you need to know both in forensic medicine and also in case of the general medicine, everything including dermatology you need to know. So this is how you see dental fluorosis, mild, moderate, severe, and severe osteosclerosis will be found. Skeletal fluorosis, Even the interosseous membrane will become calcified. That's the story. Now, when there is a arsenic poisoning, what is this called as? This is called as the raindrop pigmentation. Raindrop pigmentation is what you see whenever there is arsenic poisoning. Right, doc? And also on the teeth, you can see niche lines, diffused hyperpigmentation of the skin, etc., etc. Then, now, for a bleeding disorder and dental caries, whenever bleeding disorder is there, you need to give vitamin K supplementation. Chronic alcoholics, whenever vitamin B1 deficiency is there, that lead to development of Wernicke's encephalopathy. That is loss of coordination, confusion, etc. will occur. What is the rate limiting enzyme of the heme synthesis? It is the aminolevulanic acid synthase. This is one of the very, very important question, doctor. You need to be very sure. Now, you should know all the steps in the porphyrin synthesis, heme synthesis. Succinyl-CoA plus glycine will lead to delta aminolevulanic acid, and you require ALA synthase. And what is the related porphyria? X-linked protoporphyria. ALA become porphobilinogen by the action of ALA dehydratase. ALA dehydratase deficiency porphyria can occur. Porphobilinogen become hydroxymethylbilane by the action of porphobilinogen deaminase, whose deficiency is called acute intermittent porphyria. Then hydroxymethylbilane lead to uroporphyrinogen 3, for which you require uroporphyrinogen synthase. Congenital erythropoietic porphyria. Uroporphyrinogen 3 become coproporphyrinogen 3. It is the uroporphyrinogen decarboxylase. It is like on the Ganesh Chaturthi, we all do the Ganesh Puja. So like that, you need to remember each enzyme, what is the type of porphyria? Will it involve skin? Will it involve neurological, or both? Everything. Then coproporphyrinogen 3 to protoporphyrinogen 9. It is coproporphyrinogen oxidase, hereditary coproporphyria. Protoporphyrinogen 9 to protoporphyrin 9, which is called protoporphyrinogen oxidase. What is the enzyme? Variegate porphyria. Protoporphyrin 9 on the incorporation of the iron become heme. The enzyme is called ferrochelatase and it is called protoporphyria. Now, doctor, a neonate has hypoglycemia, hypophosphatemia, positive Benedict test. Positive Benedict test, hypoglycemia, hypophosphatemia. What is it most likely to be? It is glucose 6-phosphatase deficiency, which is called von Gehrke disease, which is the glycogen storage disorder type 1. What is the GM1 ganglioside that facilitates the entry of the cholera toxin through the intestinal mucosa? So fundamentally, whenever the cholera toxin acts, there is a GM1 ganglioside receptor. So cholera toxins EAB subunit goes and binds with that. And that in turn will lead to the sodium potassium pump to get affected. That lead to the diarrhea. That lead to diarrhea because of the CFTR receptor getting affected. All right. Now, oxaloacetate is an intermediate of TCA cycle, citric acid cycle. From where is it formed? From aspartate. So you should know, doctor, glycolysis, citric acid cycle, gluconeogenesis, every step you should know. So most of the amino acids, when they get catabolized, their atma will pass through the various intermediates of the citric acid cycle. So that is the reason you need to know about citric acid cycle. So Oxaloacetate becomes citrate, become alpha ketoglutarate, become succinyl-CoA, become fumarate. Which amino acids? Atma become fumarate, phenylalanine, tyrosine. Aspartate become oxaloacetate. Tryptophan become alanine, which will go and blend with pyruvate, then that becomes oxaloacetate. Then histidine, arginine, proline become glutamate, which become alpha ketoglutarate. All the odd chain fatty acids become propionyl-CoA. Then branched chain amino acids, valine, isoleucine, methionine, they all also become propionyl-CoA and ultimately become succinyl-CoA. So propionyl-CoA bring the atma of odd chain fatty acids that become methyl-malonyl-CoA. Methyl-malonyl-CoA mutase will convert that into succinyl-CoA. So methyl-malonyl-CoA to succinyl-CoA, you require vitamin B12. If that is deficient, then methylmalonic acid urea will occur in vitamin B12 deficiency. All right. So very simple. You need to remember these are all the crucial facts for the tomorrow's NEETPG exam, doctor. Glycolysis, where does it occur? It only takes place in the cell cytoplasm. So you should, this is a very important table. Kaisa bhi aap ratta marna padega. Mitochondria में होने वाला cycles क्या है बोलो बेटा? Citric acid cycle, oxidative phosphorylation, fatty acid beta oxidation, ketogenesis, pyruvate dehydrogenase complex, amino acid metabolism, apoptosis via release of cytochrome C. They're all the ones that happen in mitochondria. Whereas gluconeogenesis, urea cycle only partially occur in mitochondria, partially occur in the cytoplasm. साइटोसोल में होने वाले चीजों क्या है बोले बेटा? Glycolysis, pentose phosphate pathway, fatty acid synthesis, amino acid synthesis, nucleotide synthesis, lactic acid fermentation, protein translation, and partially urea cycle, partially gluconeogenesis. चल, अभी urea cycle लिए तो partially वो mitochondria, partially it is happening in cytosol. What are the enzymes of urea cycle which are there in mitochondria? Bacho, it is carbamyl phosphate synthase 1, ornithine, transcarbamylase are mitochondrial. Whereas arginosuccinate synthetase, arginosuccinate lyase, arginase, they're all cytosolic, is what you have to fundamentally remember for the tomorrow's exam. So, doctor, melanin is synthesized from which amino acid? From tyrosine. Von Geierke disease is due to deficiency of glucose 6-phosphatase. Ascorbic acid is important to convert proline into hydroxyproline. That's the reason it is required for collagen maturation. Nitric oxide, which is very important for causing vasodilatation, lead to potence. After all, sildenafil is the one which is releasing. Nitric oxide is the intermediate. So, arginine. So, you should know, arginine become nitrous oxide by releasing citrulline. Nitrous oxide lead to vasodilatation. It is anti-erythrogenic. It is anti-platelet aggregant is what you need to basically remember. So, doctor, breastfeeding is contraindicated in galactosemia. A patient with exercise-induced myoglobinuria. Exercise करने के बाद dark colored urine निकल रहा है, myoglobinuria है. What is that? It is myophosphorylase deficiency. So McArdle's disease. So you should know, doctor, the various enzymes which are involved, right? Pyruvate in a reverse way. If you look at pyruvate goes back to become glucose 1-phosphate. Glucose 1-phosphate typically is formed from glycogen. When the glycogen breaks down, for which you require myophosphorylase. If the myophosphorylase is deficient, It's called McArdle's disease. Glycogen cannot become glucose 1-phosphate, cannot become glucose 6-phosphate, cannot contribute to pyruvate and glycolysis. That's the reason myophosphorylase is very, very important for the glycogen becoming glucose 1-phosphate. A patient with diarrhea, dementia, and the photosensitive dermatitis has vitamin B3 deficiency. Vitamin A deficiency lead to xerophthalmia. In the liver, what will happen? There is one ApoB gene. It is translated to ApoB100. And in intestine, it is translated into ApoB48. All this occur because the RNA editing. So you should know, apolipoprotein B gene is the main gene. So it go to liver, it go to intestine. So in the intestine, it become 48, and in the liver, it will become ApoB100, is what you should remember. So ApoB100 is required for transport of the cholesterol in the blood, whereas the ApoB48 is required for the absorption of the lipids from the intestine. Copper, zinc, and selenium, they're all microelements. What is a complex 4 inhibitor? Cyanide is a complex 4 inhibitor. So you should know this by heart, doctor. Complex 1 inhibitor, rotenone. Complex 2 is carboxyl. Complex 3 is antimycin A. Cyanide, carbon monoxide are complex 4. ATP synthase by oligomycin. ADP translocase by atractyloside, a plant glycoside is the inhibitor. What is the medicine you use for hyperuricemia, doctor? Inhibitor of the xanthin oxidase. So you should know all the gout drugs. Nucleic acids form purines, purines form hypoxanthin, become xanthin, become uric acid. Xanthin to uric acid, you require xanthin oxidase, and that is inhibited by allopurinol and febuxostat. Uric acid typically become allantoin, which is a water-soluble metabolite that is promoted by urate oxidase, which is stimulated by Rus buricase. You should remember. Similarly, how will uricose-uric drugs will act? Like sulfinpyrazone, probenecid, they will make the uric acid to become excreted through the kidney, is what you need to remember. So now, doctor, a child with urine turning black on standing typically has alkaptonuria. Vitamin B12 deficiency lead to subacute combined degeneration of the spinal cord, where the myelin around the nerves is gone. That lead to the tingling sensation. What is the enzyme deficient in Tay-Sachs disease, doctor? Hexosaminidase A is the one which is deficient in Tay-Sachs disease. So you should know everything, anything about all these things, doctor. GM2 become GM3, for which you require hexosaminidase A, Tay-Sachs. GM3 become glucocerebrosidase becomes ceramide. For that you require glucocerebrosidase deficiency lead to Gaucher's. Similarly, ceramide also can become glucocerebroside, for which you require alpha glucosidase A, and the deficiency of that is called Fabry. Then ceramide is also formed from sphingomyelin, for which you require sphingomyelinase. Deficiency of that is called Niemann-Pick. Then sulfatides form galactocerebroside, for which you require aryl sulfatase A. Deficiency of that is called metachromatic leukodystrophy. Galactocerebroside becomes ceramide, for which you require the galactocerebrosidase. There's different glucocerebrosidase and galactocerebrosidase. Galactocerebrosidase deficiency is called Krabbe. Whereas the glucose ribrosidase deficiency is called Gotcha, is what you all need to remember for the tomorrow's upcoming exam. Now, doctor, when the mother's side uncle, mama, and her son both have the same disease, mother side uncle and her son, mother is there, her brother and her son, both have the same disease, means it is X-linked recessive. So you should know how to recognize the pedigrees. Sex-linked dominant. Sex-linked dominant. All the females are the ones who get affected. Right? And female can cause it to pass to her son. Autosomal dominant. Autosomal dominant, how you identify? Similarly, sex-linked recessive and autosomal recessive. Which get vertically transmitted, autosomal dominant. Which get horizontally transmitted, autosomal recessive. Now, the medicine for raised uric acid, once more repeat, xanthine oxidase inhibitor, like febuxostat or allopurinol is what you have to remember. Nitric oxide is synthesized from arginine. These are all 6,500 question bank of the FMGE past 10 years. Chaperone, protein folding. Alcoholic patients with neurological symptoms is vitamin B1 deficiency. That's the reason always whenever you give glucose in a hypoglycemic patient who is alcoholic, don't forget to supplement the thiamine. Otherwise, the patient will end up in beriberi. That's very, very important clinical practice. May you find a lot of alcoholics coming. Whenever they come in hypoglycemia, when you are giving glucose, add thiamine to the glucose. A patient has macrocytic anemia, homocystinuria, so he can benefit from vitamin B12 therapy. So you should know, doctor, everything, the funda behind it. Methionine is formed from homocysteine, for which you require methionine synthase, which has vitamin B12. But methionine synthase also converts the 5-methyl tetrahydrofolate into tetrahydrofolate. So if there is no folate, also methionine synthase cannot occur. Homocysteine will be excessive. It can't be converted into methionine. That's the reason there is a homocystinuria. Similarly, any vitamin B12 deficiency is there also, then that can lead to development of homocystinuria, is what you need to remember. The isozyme LDH1, it is elevated in the heart conditions. So what is the cofactor for the mitochondrial superoxide dismutase? You should know all the trace elements and where are they, the cofactors. Manganese. So superoxide become hydrogen peroxide, that become water. Superoxide to hydrogen peroxide, you require superoxide dismutase. Copper, zinc, manganese are the cofactors. Hydrogen peroxide become water, catalase, iron, glutathione peroxidase, selenium. Any of them can convert hydrogen peroxidase into the water. Choline, folic acid, methionine, they are given in the fatty liver. Choline, folic acid and methionine. So folic acid is given because for methionine formation from homocysteine, you require vitamin B12 and folic acid. Right, doctor? Then choline become betaine. Betaine become dimethylglycine. Right? When the homocysteine is becoming methionine. So that is the reason choline is also administered in liver disease. The first fatty acid to form in the fatty acid synthesis is 16 carbon palmitic acid. Nitric oxide is formed from arginine. In maple syrup urine disease, what is deficient? Branched chain alpha keto acid decarboxylase is deficient. What is the most abundant amino acid in collagen, doctor? Glycine. Vitamin E is the most potent antioxidant, is the vitamin E. For the iron to get absorbed, you require which vitamin, doc? Vitamin C. Wernicke encephalopathy, thiamine deficiency. Then, what are the various uncouplers? Just before we studied thyroid hormone, unconjugated bilirubin and long chain fatty acids are all uncouplers. Now, what are the enzymes in glycolysis? Phosphofructokinase, glucokinase, pyruvate kinase, they're all the enzymes. So quickly tell me, doctor, what are the various steps in glycolysis? Glucose become glucose 6-phosphate hexokinase. Glucose 6-phosphate to fructose 6-phosphate phosphoglucoisomerase. Fructose 6-phosphate to fructose 1,6-biphosphate phosphofructokinase 1. Fructose 1,6-biphosphate become dihydroxyacetone phosphate and glyceroldehyde 3-phosphate. You require aldolase. Between the two, for the conversion, you require triose phosphate isomerase. Glyceroldehyde 3-phosphate become 1,3-diphosphoglycerate, glyceroldehyde 3-phosphate dehydrogenase. 1,3-diphosphoglycerate become 3-phosphoglycerate. Typically, it is phosphoglycerate kinase. 3-phosphoglycerate become 2-phosphoglycerate become phospho. Enol pyruvate become pyruvate. So all these steps, where is ATP produced, where is substrate level phosphorylation, everything you should know, doctor. Anderson, Palm, Von Geijk, they all will affect the exercise. Because at the time of the exercise, you get cramps, etc. McConnell doesn't. So this is very important table, doctor. How do various glycogen storage disorders, what are the enzymes? Von Geijk, glucose 6-phosphate. Palm, acid alpha glucosidase. Corey, debranching enzyme. Anderson, branching enzyme. McConnell, muscle glycogen phosphorylase. Hirsch disease, liver glycogen phosphorylase. Tarui disease, phosphofructokinase in the muscle. Fanconi-Bickel is glucose transported to GLUT2. In that, what are the clinical features you should know? So this is, every time you go to the NEETPG, this is the favorite area for the examiner. Gluconeogenesis, mainly where does it occur in the liver? Whenever glucose 6-phosphate dehydrogenase deficiency is there, it leads to the hemolytic anemia, G6PD deficiency. Lack operon is induced by decreased glucose concentration and increased cyclic AMP. The lack of operon is being induced. So what constitute creatinine? Glycine, arginine, methionine, glycine, arginine, methionine, they constitute creatinine. So glycine and arginine combine to form guanidoacetate by releasing ornithine. Guanidoacetate typically become acetinosyl homocysteine. In the process, acetinosyl methionine provide acetinosyl group. Methionine become homocysteine. Right? So SAM, all methylation reactions, SAM is the methyl group donor. So guanidoacetate become creatinine, and what is that enzyme called? Guanidoacetate methyl transferase. Now, doc, amyl nitrate is an antidote for cyanide poisoning. It is an example of chemical antagonism. So you should know, whenever cyanide poisoning is there, what is the main damage, cytochrome oxidase is inhibited, right? So hemoglobin is converted to methemoglobin by the nitrate, which has a high affinity for cyanide, and that becomes methemoglobin with the cyanide. Then from that the cyanide is being dropped off, that combines with the thiosulfate, leading to the excretable form of cyanide, thiocyanate, and goes into urine. So that is the reason to convert hemoglobin to methemoglobin, you use nitrate. Methemoglobin sucks the cyanide, and then thiosulfate will detoxify the cyanide into thiocyanate and excrete into urine. Type 1 collagen requires vitamin C. So that brings us to the end of today's wonderful evening, doctor. हम रोज मिलेंगे, रोज पढ़ेंगे, and one day we will become the winner. So be part of the WhatsApp study group, like this video, share this video, and be the part of our WhatsApp study group. Thank you very much for joining with Dr. Murali Bharadwaz, your everyday classmate, roommate, benchmate, table mate in your NEETPG preparation. Good night.